Hi, my name is Kyle Hippe and I'm with Pacific Lutheran University, and today I will be presenting CaoLab Prolango 2, which is a protein function annotation method from a sequence-based RNN. So first I would like to describe the protein function prediction problem. This problem states that given an unseen protein, we must determine a predicted set of biological functions. Currently we must do this with biological experiments, and there's a couple issues with this. The primary being it is very resource and time intensive. So a computational method would help us bridge the gap between the relatively small number of annotated sequences and the increasing amount of unannotated sequences. So to solve this problem, we would like to introduce Prolango 2. So a brief overview of Prolango. Prolango is a novel character-level RNN based on a neural translation machine. This is also an ensemble method, so we take the top 100 performing models and combinatorially analyze them for the maximum prediction accuracy. In the end, we have 20 models that are used in the final ensemble, and we will go into a bit more depth about how we create these models. So first, we must consider the protein sequence as a language. So if we consider a protein sequence as a sentence, then if we draw corollaries to human language, then a sentence consists of words. In the context of a protein, a word would be a substring of amino acids. To develop our protein language dictionary, we analyze the UniProt database consisting of 536,000 proteins, and we search for amino acid substrings ranging in length from 3 to 7. Our training set could then consist of the top 2,000 occurring of these words, which then constitute our protein language. So as you can see in this example, we take a protein sequence and we split it up into its protein words. So since this is a translation machine, we must have an output language. And the output language in this case is the functions of the protein, or the gene ontology terms. So we must consider these as a language as well. So since gene ontology terms are three directed acyclic graphs representing the biological processes, molecular functions, and cellular processes, we perform a depth-first search on them to get a base 10 index for each term. Once we have this index, we can convert this base 10 into base 26, or a letter representation, and each of these would be a maximum of 4-length string, which represents or directly correlates to the Go ID of the Go term. So if we have a gene ontology tree that looks like these, we have molecular function, biological processes, and cellular components. We perform a depth-first search on these to get indexes, and then once we convert those into letter strings, then we get a gene ontology tree that looks like this. Each of these terms directly correlates to the IDs, and they now represent our gene ontology language. So now that we have both of the languages, it's now time to move on to training our RNNs. So in the salmon color boxes, we have the determining the protein language. So once again, that's converting or finding the top 2,000 occurring words from the UniProt database. In the blue colored boxes, we have converting the Go IDs into Go words, which are the 4-character strings. And now we move on to the encoder and decoder of the neural machine translation part of our method. So the encoder is a sequence or a character level RNN that receives the protein words from a input sequence. Then it encodes these words into a content vector. From there, a decoder RNN, a completely separate RNN, receives the values from the content vector and then predicts the terms in the new language, the Go language. Now that we have our Go word predictions, since we can directly translate these Go words back into Go terms, and thus we have our predictions. Now, like we said earlier, this is an ensemble method. So this just represents one of our RNNs that we have in our final ensemble. And as we said earlier, there are 20 RNNs in our ensemble, so there would be 20 of these that are making predictions for each sequence that is given to us. Now for a bit of results. We tested 253 unique sequences taken from the UniProt database, and we tested these using the top N metrics, which provides statistics for predictions ranking in ranges 1 through 20 for each target. We analyzed the maximum similarity and the average similarity of the Go terms in the predicted set to the Go terms in the true set. So here we see the targets or the methods in blue are similarity based, the methods in green are network based, the the methods in red are sequence-based and the methods in black are information-based methods. And as we can see for the maximum similarity, Prolango performs especially well for sequence-based methods, but has some ground to make up for the network-based and similarity-based methods. And moving on to the average similarity, Prolango struggles here. It performs poorly compared to sequence-based methods and has a lot of ground to make up in comparison to the network-based and even more to make up for in the similarity-based. Even despite these pitfalls, we believe that we can improve these with future work. Some of the future work would be including different models in the ensemble. We've considered hidden Markov models and Bayesian neural networks. Second, since this is an ensemble, we could tune the RNNs for the molecular function, biological processes, and cellular components. These sets of models would be only trained on one category, and then we could combine them to give very accurate predictions for each category. Second, we would also like to incorporate similarity-based methods for prediction when homology is high, and then relegate the RNNs to novel sequences where homology is low. For acknowledgements, I would like to acknowledge Dr. Renzi Cao for his patience and support, Shola Benro for his support as well. We would also like to thank the Google Cloud platform for their resources and training, as well as the NSF and PLU for their both monitory and resource support of us. For implementation details, please visit our GitHub at the link provided. For the previous Prolango publication, please visit the PubMed link provided. And for any questions or comments, please direct them towards myself, Kyle Hippe, at the email link provided, or Dr. Renzi Cao at the email link provided. Thank you very much for listening,